Okay, so let's get started. I thought maybe there's some familiar faces in the room, but not many. I'll just give you a brief introduction to who we are and what we try to do in my group. So we've long had a focus in understanding the different mechanisms that contribute to enabling large groups of cells to migrate as a coherent sort of collective, as a coherent entity, often over really quite large distances. And then how these mechanisms combine with other kind of things that a cell might do, like proliferate, so divide, or to die, to give rise to very complicated biological phenomena. And we have traditionally worked at the kind of at the embryo development end of the scale, really with a view to thinking about, if I can understand how a tissue is formed de novo as a result of this collective migration of a population of cells, then that will enable me to have a platform to think about sort of maintaining tissue, so homeostasis, but also various pathologies. So, for example, tumor growth arising as a result of uncontrolled cell proliferation and migration, understanding what happens when things go wrong and we need to impact wound healing, or thinking about how to generate new tissues in a laboratory context. And what we try to do is we try to combine increasingly these sort of mechanistic mathematical statistical physics models with other techniques from statistics and machine learning to provide new biological insights. Okay, so today what I've tried to do with my talk is intentionally keep the biology quite simple, keep the math quite simple, and then try to focus on what we can learn when we combine the two. And also to try to kind of maybe make a few points about what we might consider along the way in terms of building our mathematical models. So I'm going to ground everything in mostly the data and the biological problem that we're interested in. So I said it's all about collective cell motility. What I want to think about today is essentially how the kind of phenomenon that is cells migrating as a collective, and so we've got a large population of cells, might influence essentially how cells proliferate. So this is sort of feedback between proliferation and migration, if you like. Okay, so how do cells proliferate and what do cells actually do? I don't think I'm tall enough, even with the pointer maybe. We'll see. Okay, so you can a very basic level. Okay, and apologies for the kind of biology people in the audience for me breaking this down. So simply, essentially, a cell over its lifetime will go through the cell cycle many times, right? And we can break the cell cycle down roughly into 4 phases. So G1 is the sort of first phase that starts just after division, okay? And then after G1, a cell enters what we call S phase, and then G2 phase, and then eventually M phase, which is called mitosis, and that's where it's going to divide. And then when the cell divides, the whole process sort of starts again. So that's a very kind of coarse representation of the cell cycle. And what I really want to know or understand is how the cell microenvironment, and in particular, how does sort of population and crowding from other cells influence the progression of the cell through its cycle? That's my big question, if you like, for today. And so I'm going to try and build some models and compare the models with data to try and answer that question. Now, one of the reasons that we thought we could begin to get a handle on that question is because, almost 20 years ago now, a new technology was developed. And that technology is known as a feature reporter. So what a feature reporter does is effectively you engineer it in a cell, and so the cell will make a red fluorescent protein when it's in G1 and a green fluorescent protein when it's in the rest of the cell cycle. And so when you kind of image the cells, what you can see is a very coarse representation or coarse information on where a cell is in its cycle. So largely speaking, in G1 it's red. You've got a brief period where both of those fluorescent proteins are around and a kind of crossover between G1 and S, and the rest of the time it'll appear green. And you can engineer that reporter in sort of cell lines and do experiments in the dish, which is what that left-hand figure is. But you can actually put it into entire organisms. So the kind of things that look more like an entire organism on the right there, I think that's a developing embryo. You can see that across the embryo, there are these huge patterns of coordination of the cell cycle. So anything that's green is cells all in SG2M, for example, and they're maintained over time. So I am really interested in, now how do I start this movie? Oh, here we go. I am really interested in using data that looks something like this to understand how the cell cycle gets regulated by local sort of crowding. And just to, you can all see, but just to illustrate, we can really see these clear patterns of after division, cells being red, they're sort of transiently yellow, they go green, and they go dark. And that's the kind of data that I'm going to use throughout my talk. So I want to understand, if I built some mathematical models that incorporate these kinds of cell cycle dynamics into them, can I learn, and I've said this already, how the cell cycle is regulated through crowding? But I've got a lot of questions that I want to kind of ask along the way. So as a mathematical modeler, one of the things we're always interested in asking is my model at the right level for the data? So essentially what I mean by this is it shouldn't be so simple that I don't learn anything new, but at the same time it shouldn't be so complicated that it can basically tell me anything I like. So there's a sort of sweet spot in the middle, and we've long kind of said that this is a real talent and of an applied mathematician to think about what that right level of the model is. And I'm going to sort of try to highlight today that I think we can use some sort of less heuristic methods to think about, is my model at the right level of the data? And that's by asking the question, are the parameters of my model identifiable? And I'm going to spend quite a long time talking about this today, but really that just asks the question, given data that I've got, can I estimate the model parameters, right? And in doing so, I'm often interested in kind of how certain those estimates are, right? So can I quantify the uncertainty at the same time? And the reason that that is important, because then it allows me to ask the next question, if you like, which is how does any uncertainty I have in those parameter estimates impact the predictions that I then might make with the model in new scenarios? So if I want to go to one of my collaborators and say, can you do a new experiment that I want you to test a kind of hypothesis that I've got, okay? So this kind of idea of quantifying uncertainty allows me to think about what are the range of possible outcomes that I might expect from that experiment and think about essentially whether I can, in fact, essentially see something that's consistent with my hypothesis or falsifies it. Okay. I'm going to say something briefly, though, today, I think, about how changing my experimental setup might have an impact on my ability to estimate parameters. And then I'm going to try and I will talk a bit about the biology and what we learn after we thought about all this. Okay. So what does the data that I'm going to work with today look like? It's data, and hopefully you can see this, that's collected from two different types of experiments. One in the lab of a collaborator, Daniel Cohn from Princeton, and the other in a lab of a collaborator, Matt Simpson from QUT. So these are simple experiments. We like simple because we're still learning how to do this. These are simple experiments that are carried out in vitro in a dish. And so the top experiment, so you can see this experiment run over a period of about 40 hours. This one up here is what's known as a barrier assay. And so what you do, essentially, is you get a population of cells and you grow them inside what I would call like a cookie cutter, effectively. And you grow them for a while until they reach some kind of fairly uniform confluent density. And then time zero of your experiment is taking that cookie cutter away. So that when you do that, the cells can move into the available space and they begin to proliferate. Okay, or carry on proliferating if they already were. And then the second type of experiment is called a scratch or a wound healing assay, where you grow this time the cells to population to confluence in the whole dish before you start. And then time zero is kind of you taking a pipette or a sharp-tipped instrument and essentially just scraping away a portion of the cells down the middle of the dish. So that when you take an image, what you see is that the cell population, these are red and green cells, are sort of on either side, and you've got this hole down the middle. And again, as you watch over time, what you can see is that the cells move in from the sides, they proliferate, and they fill the available space. So this is the data I've got, and it's captured with varying degrees of temporal resolution. But I'm just showing you a few snapshots through here at the moment. But I'm generally going to assume that I can't actually track individual cells, so I've just got snapshots of where the cells are at any given time. Okay, so what's my first question? My first question is really kind of what parameters of a model can I hope to be able to estimate with these kinds of data? And today, I'm not always wedded to this, but today I'm only really going to talk about partial differential equation models. So when I say what parameters of a model, they're parameters of sort of generally parabolic reaction diffusion type PDEs or systems thereof. And I'm often going to think about either modeling the evolution of the total cell density over time or the density of red cells and the density of green cells and how they transfer between the two. So when I ask the question, what parameters of a model can I hope to be able to estimate using these kinds of data, one of the first things I might think about is a concept known as structural identifiability. So I'm not an expert by any means on this, so I like to break it down into kind of a simple question. So structural identifiability is all about asking, do distinct parameter values in my model give rise to distinct parameter sets in my model give rise to distinct distributions of observations? That's really what the question I'm asking here. So structural identifiability has nothing whatsoever to do with the data, right? But it's a property of the model, okay? And really, if the answer to this question is yes, right, that means that it's possible in theory, given perfect input-output data, to be able to determine the values of my model parameters from data. So if I have perfect data, and this is true, I can estimate the parameters. Okay, so it doesn't take into account, the limitations of the data, which you've already seen on the previous slide is noisy, and it's incomplete in the sense that I don't sample right in continuous time. But it's a very relevant first question to ask, simply because if I can't do this with perfect data, I can't do it with the real-life data. Okay, so I want to tell you a little bit about some progress we've made in determining structural identifiability for partial differential equation models. But I'm going to start by telling you about how you might go about it for ordinary differential equation models. So I've got a model here that's just two species, Q1 and Q2. It's a bit of a toy model that people use in biology. It's not particularly relevant what it does today. But what do I want to say? So I've got initial conditions for Q1 and Q2. I've got a control input, which is U, which is some time-varying input that I can change. And I've got four parameters in my model, P1, P2, P3, and P4. Okay. And so the question is that in the context of what these Q1 and Q2 mean or are, then all I can actually measure are observations of a quantity that I've called Y, and Y is Q1 divided by some VP, which is a representative volume that I also might not know. So I've got a number of parameters in my model. So a sort of differential algebra approach to understanding structural identifiability says, well, what you need to do is you need to write your model, so your two differential equations, in terms of this observed variable. So you do some differentiating and substituting, and what you get is a higher order, so a second order in this context, ordinary differential equation that tells you how Y changes in time. Okay. And so what's the idea here? So this is not always a particularly simple thing to do, but once you've got here, the fact that you've got perfect observational data of Y in time means that you can essentially, you can measure all these derivatives, or you've got access to all of these derivatives. Okay, so as a result, if this thing is now monic, right, that's important, so there's at least one coefficient that's 0. What I can see is that if I read off the coefficients of that differential equation, they are essentially a set of identifiable parameter combinations. So here what you can see is that as long as U is time varying, I should be able to get P3 from this term. I can get VP from this term. And then this term tells me that I can get P1, because I've got VP and P3. And then when I look at this term, what I see is that I can then get P2, P4, right? I can get the product of them. So what this sort of very simple analysis tells me is that it doesn't matter how good the measurements I have, right? The only kind of things that I can ever hope to estimate are P1, P3, and the product of P2 and P4. Okay, so this is an approach that's really well established for ODEs, and what we wanted to understand is whether it could be extended for partial differential equations, because up until this point there wasn't really any work that had allowed people to determine structural identifiability for partial differential equations. So we asked a super simple question, can you just extend it, right, to PDEs, and we're interested in reaction diffusion advection PDEs. Okay. So to show you how it works, I'm going to ground everything in an example, and it's going to be an example that I might use to describe this experimental data. So one of the things you can do quite nicely with one of these scratch assays is you can average all the data in the y direction, right? Remove a degree of complexity, if you like, so that essentially you have a PDE that's a function of average density in x and over time. So it's simpler and hopefully a little bit less noisy as a result of that averaging in y. And so I've got data which is essentially sort of density of cells in G1 in red and density of cells in SG2M in green. And the simplest possible model that I could write down for this system is this one. So it just says that the density of red cells, again, my height's like, the density of red cells evolves over time because these cells move randomly with some diffusivity D1. And then I see a loss term in this equation because this parameter K1 is the rate at which cells essentially are transitioning from G1 into SG2M. So there's a plus down here. And then I get a plus K2G up here because at the end of the cell cycle, my cells in SG2M divide to give me two cells back in G1. So it's about as simple as it gets. And just to highlight that the green cells also move randomly, and then they have the kind of complementary kinetics, if you like. So a question you might want to ask is, do I really, so what I want to be able to do, right, in theory, is to estimate these four parameters, D1, D2, K1, and K2. And in particular, I guess it's K1 and K2 that tell me something about how the cell cycle, or the length of the cell cycle. So here I'm ignoring anything to do with cells crowding one another and that impacting the cell cycle dynamics. I'm just saying they don't see each other, they march through the cell cycle, disregarding each other. And so 1 on K1 is the average amount of time a cell would spend in G1, and 1 on K2 is the average amount of time it would spend in the rest of the cell cycle. Okay, so where was I at? Okay, so you might ask the question, do I really need this reporter, right? Because those pictures that I showed you look absolutely amazing, but when we've tried this in a cell line, a cardiac fibroblast cell line in oxids, it was really hard to use, right? And the levels of fluorescence aren't the same between the two phases of the cell cycle. So although those pictures look pretty, it's not necessarily the easiest thing to collect data with. Okay. So we said, actually, do we really need to measure both density of reds and greens, or can we just use something like a bright field image, right? So just take a photo and look at the total cell density. And can information which just has total cell density, is that enough to enable us to estimate all these parameters? So what I want to do is do exactly the same thing as you saw before, reduce the model to an input-output equation, and now it's going to be in terms of this observed variable n. And then the coefficients in this equation will tell me my identifiable parameter combinations. So I've used a slightly compact notation where nij now is the ith derivative with respect to x and the jth derivative with respect to time. And so you can differentiate and substitute. Essentially, first of all, get rid of, which was I was going to say, first of all, get rid of the reds and then eliminate the greens to get this equation here. Okay. And then you read off the coefficients and you get this set of identifiable parameter combinations. And if you look through that, you can see that, I guess, under the assumption all the parameters are non-negative, that essentially I should be able to estimate all of my model parameters. So I should be able to get K1, K2, D1, and D2 just from this cell density level data. So this model is structurally identifiable. Okay, so if that were the whole story, it'd be really great. And I'll come back to why it is in just a second, isn't in just a second. But I want to say that in this paper where we talk about this, we extended this to kind of more general reaction, diffusion, advection systems. So if I give you a slightly more general equation of the same sort of type, where now I've got components U and V that diffuse, advect, and then there's some linear kinetics which they convert between U and V, and then there's some source or sink terms P3 and P6. Okay, I can ask the question in this more general context, and if I only measure the sum of the two, what can I do? Well, you get your input-output equation, and it's quite a bit more work. But when you get there, you can read off the set of identifiable coefficients from this equation, and here's what they are. So it looks a bit messy, but then what you can do is use some of the techniques that the ODE people have been using for differential algebra for ages, and you can find essentially what is the sort of the reduced set or the minimal set of identifiable parameter combinations. And what this tells you, actually, is that if you do just measure the total density, you can get both the diffusivities, you can get both the advection coefficients, or terms, and then you can get some information about these kind of rate parameters. So you can get P1 and P5 individually, and like similar to our previous one, you get the product of a couple of parameters. And then I think for P3 and P6, which were those sort of source or sink terms, you can just get them in combination with many of the rest. So you can do it and you can turn the handle. The one point I want to make about this is that if you then assume that you were conducting an experiment with U and V, and you set that experiment up in such a way that there were no spatial gradients and there was no advection, so set the Ds and the alphas to 0, then you get a different set of identifiable parameter combinations. And the thing that I think is really interesting is that it's actually worse. So I can't get any of the individual rate parameters out of this. So it really says it's a first clue that experimental design really matters. If I do an experiment where I initialize it in the spatial gradients, or I'm using some microfluidic setup and I have a flow, then I should be able to do much better in terms of estimating my model parameters than if I do a simpler experiment where everything is well mixed. Okay, so that was my slight detour. Let's come back to the problem we're thinking about. So I was talking about the fact that this kind of sort of base level sort of analysis indicates that I don't really need this Fucci reporter. If I just measure the total cell density, I should be able to get all the parameters in my model to estimate them. And you could ask the question, what have we missed? Well, what we've subtly missed is that things get more complicated in going from ordinary differential equations to partial differential equations, because instead of our initial just a condition just being a constant, right, it's now an unknown function. So when I think about having n, which is the sum of, in our case, R and G, or U and V in this context, I don't necessarily know kind of what proportion of my total density is in the U state and what proportion of it is in the V state, right? And so I've got this initial function that I don't know. And so this is essentially my input-output equation evaluated at the initial time point. And what I want to kind of just say, and I'll highlight this with an example in the moment, is that if you sort of make almost unwittingly make assumptions that you know, right, what the initial condition is for V or the initial condition for U is, then what you can do is sort of unknowingly render a problem as looking structurally identifiable when it actually isn't. Okay, so you have to be very careful in kind of what you assume you know initially. To show you that's the case, here I got a set of examples where I've got that diffusion advection reaction system with some fairly arbitrary parameters. And the top line is one set of simulations with one parameter set, and in the bottom row, I've just literally doubled one of the parameters. And by a careful choice of change in the initial condition, right, I can give rise to output from the total density that looks identical. Right? So essentially, we need to be really, really careful in drawing conclusions about structural identifiability if we don't know what the initial conditions are, so the initial separation of the two populations. So what that actually says is, going back to the problem I'm interested in, unless I know what the density of red cells is at every point initially, right, my problem isn't actually structurally identifiable. So I do need this victory reporter after all. Okay. So if you're interested in this, you can look at the paper, and there's some code that helps you do this. All right. I'm going to keep going because I spent more time talking about that than I thought I would. So where have we got to? We've got to the fact that we can write down a PDE model of this system, and that we really need this feature reporter and data from it in order to be able to estimate the parameters of our model. Okay. So now I want to talk about a notion which is slightly stronger, which is the notion of practical identifiability. And that really asks the question, given the data I actually have, is it possible to provide reasonably precise estimates? So it's a little bit of a woollier concept, if you like. But really, if I have the data, how well can I do? Can I estimate the parameters? And then can I estimate them kind of with a better degree of confidence than I already think? Okay. So I want to ask the question, can we build a practically identifiable model that includes cell cycle dynamics, and what can we learn about regulation of the cell cycle given this kind of data? And so the model I showed you before had no cell cycle regulation in it because cells were just transiting from one part of the cell cycle to the next, irrespective of the cell density. And I want to just make that model slightly more complicated to take that into account. But first of all, one of the things to just highlight is that one of the great things about working with Daniel Cohen and his group is that they've got great kind of image analysis technologies where they can literally automatically detect every cell in any one of these images and tell you whether it is in G1 as red, SG2M as green. And these gray cells are cells that have just divided, so they don't have either expression. And we use that to effectively get a one-dimensional cell density profile. So this is just kind of radially averaging out from the center of our colony. So what do our models look like? Again, they're systems of reaction diffusion equations. So I'm going to lump these gray cells, the post-mitotic ones, in with the G1 cells. So they're all going to be called red cells. And then I've got density of green cells as well. So, and I can talk about this another time. I'm going to assume that they both diffuse. There's the same diffusion coefficient for both populations. And then I've got these same dynamics where at the end of being in G1, cells go into SG2M. And when they divide, that gives me this factor 2 up here. But the difference in my model now, and it's really still super simple, is that I have these terms in here, F1 and F2, which just capture the fact that these kind of basal rates of cell cycling can be disturbed by the kind of the local cell density. And in fact, what I anticipate is that the cells are going to cycle more slowly as the local cell density increases. So we've got these terms in here that essentially are crowding-based regulation of transitions between phases. And because this was a really initial study, we just took the simplest possible form where the rate just decreases from 1 to 0 as the cell density increases up to, in this context, up to a density K1, capital K1, and in the other context, up to a capital K2. So essentially what this says is that the rate of transitions from G1 into SG2M decreases linearly, and it becomes 0 whenever the cell density hits K1 or goes above it, and the same in the other context. So the question is, can we estimate the parameters of this model given the data, and are they practically identifiable? So do we have a good degree of confidence in our estimates, and what's the uncertainty in them? So if we want to think about uncertainty in parameter estimates, we're going to want a Bayesian framework. So we're targeting the posterior probability of our parameters theta given our data d, and we access that through the product of the likelihood and the prior. And we tend to assume for the prior distribution like very little kind of knowledge, right? So you use some uniform prior between 0 and some feasible upper bound for any one of these parameters. And then properties of that posterior are going to give us information about the parameters and also our inferential precision. So we might be thinking about looking at things like the 95% credible interval. So to connect model with noisy data, we need a noise model. So we're just going to assume that the data that I have, right, comes from simulation of the model with a given set of parameter values, and there's some observation noise, which is IID Gaussian. And that's a very standard thing to do, and you can change that and make it more or biologically relevant if you want. But the nice thing about that is it gives you a very easy to compute likelihood, because as long as you can solve the PDEs, then you can just evaluate the likelihood of the data given the model. Okay. So what I'm showing you here is just the outcome of using an MCMC algorithm very simply to just target those posterior parameter distributions. And so these are the marginal posterior distributions for the parameter values. And so this is essentially for the diffusion coefficient. And you can see that it's fairly tightly constrained to lie in what we know to be fairly sensible values. And then you can get estimates of both the rate parameters K1 and K2, which are these basal cycling rates, and the carrying capacity. I'm going to call them parameters capital K1 and capital K2. Okay, so is the model practically identifiable? Yes, we've got pretty confident estimates of these parameter values. And the nice thing is that we can assess the uncertainty too. The other thing that you want to go away and check is that actually your model does a good job at predicting the data that you have. So in the squares, you can see is the average results from 11 different replicates of the experiment, and then the ribbons are essentially just the variability on that, standard error. And then the solid lines are just the results of simulating the model forwards in time using the maximum a posteriori estimates. And what I want to highlight is a few things, actually. Number one, I think it does quite a good job. But in particular, what it does is a really, really good job of capturing the dynamics around the edge of the colony. And in particular, this kind of local sort of peak, if you like, in the density of cells in SG2M, just behind the front. So it's doing a really, really good job at capturing the data. So it's great. So one of the things that you can do then when you've got these parameter estimates is then you can begin to get a sense of how the rate at which cells cycle depends on their local cell density, right? So these are plots that just show us on average, how long will a cell spend, for example, the red line is how long it will spend in G1 or post-mitotic as a function of the local cell density. So the red line is the mean, and then we've got some sort of uncertainty that's quantified by the gray around that. And then I can do the same for the second part of the cell cycle in green, and adding them together just tells us something about how long the cell cycle in its entirety is predicted to be as a function of the local cell density. Okay, so there's a couple of things I wanted to say about this. Number one, does this make sense, right? And I'm going to go back a slide to talk about that this might make sense. And in particular, I'm interested in these carrying capacity parameters, capital K1 and capital K2. So K1 is smaller than K2, right, in our predictions. That's what's coming out of the data. Is that sensible? Well, it is, I think, actually very sensible, because this is a checkpoint. And if you go back, I wish I'd got maybe I can do some drawing on the board, maybe not. If you go back to what's going on in here, it's busy. K1 is the checkpoint at the end of the first part of the cell cycle, right? And what this is telling us is that checkpoint is actually a stronger checkpoint than the checkpoint that kind of is at the end of the cell cycle right before division. And that makes sense, right? Because there's in that second part of the cell cycle, SG2M, a cell is going, making all the extra sort of material, increasing its volume, ready to divide. And this tells us that it's not going to do that unless it's got a fair degree of confidence that it's actually going to be able to divide at the end of the cell cycle. So making that first checkpoint stronger is, in fact, a really, really sensible thing to do for a cell from, if you like, an evolutionary perspective or an energetic perspective, maybe. Okay. The other thing you might ask is there actually any evidence biologically for these checkpoints to exist in biology? Well, we've known that there's one of them for quite a long time. So this is some work coming out of Lars Hufnagel's group, I think in 2014. And they established, using the same type of cells, essentially that there is a mechanical checkpoint at the boundary from G1, so the end of the first phase of the cell cycle, into G2, which is or S, which is the second phase of the cell cycle. So we were confident that that existed and that our model, in fact, is picking up that it exists. But the thing we really weren't confident about is the other one. So when we were doing this work, we weren't aware of any evidence to say that there was a second checkpoint in the cell cycle. And so Carlos, the student who was doing this work, was a bit worried, and I said, it's all right, let's just go and do a bit of a literature search. And it took a while, actually, to uncover this. But in fact, there was some very recent work relative to the time when we were doing this, coming out of Martin Glorich's in Utrecht, that actually provides evidence for a mechanical G2 checkpoint, so the second checkpoint right at the end of the cell cycle. So we picked it up from basically having a model and needing that model to fit the data, and they'd independently established it through a series of experiments. So it's really nice to see that there was evidence for that. Okay, what else can you do with this model? Maybe I'll just say a couple of things about that. You can do a very, very standard traveling wave analysis that we teach all our undergraduates in third year or something. And the nice thing about that is that allows you to establish an estimate of the wave speed in terms of the model parameters. So here in particular, the wave speed is predicted just to depend on those cycling rates K1 and K2. And that's precisely because this is a linear analysis and all of those kind of higher order terms that contain the capital K1 and capital K2 disappear. So the wave speed is predicted to be independent of the extent of crowding. But on the other hand, if you do some numerical simulations where you vary these capital K1 and K2 and you vary the initial condition, what you can see is that not only does the shape of the wave, but the composition of the wave, this invading population, depend hugely on those parameters. So you can really kind of just switch the composition and the makeup of the wave by playing around with your initial condition and those two different parameters. So there's a whole spectrum of behaviors that you could expect to see in biology. Okay, so I've got a few more things to say, but I'm going to give you a bit of a take-home message and then say them. So what I've tried to hammer home today is a bit that I think, considering both structural and practical identifiability, is really important when building models. And one of the things I would like to sort of push for a little bit is considering or using practical identifiability as a way in which to think about what level of model is right for the data. So we kind of remove some of the heuristic nature of choice of model. Okay. I've hopefully also maybe showed you that ODE-based methods for structural identifiability can be quite easily extended to partial differential equations. And actually, I think one of the biggest challenges lies in deriving those input-output equations. So we showed that you can always derive them as long as the thing that you're shooting for is linear in the observed variable. But doing that in fuller generality is tricky, and it takes a very long time. So I think the key to kind of making progress for structural identifiability for PDEs is really going to rely on having good software that can generate those input-output equations, as it was, I think, for a long time for ODEs as well. So I said I'd like to lean on practical identifiability a lot more in the process of building models, and I think concepts to do with sort of profile likelihood analysis, for those of you that think about these things, I think can be cheaper alternatives that allow you to, in fact, assess practical identifiability without computing entire posterior distribution. So my model was quite simple, and MCMC works quite quickly, but for larger models, that isn't always the case. I also want to say, and what I showed you as well is that quantitative data allows us to say something about the extent of density-dependent regulation of the cell cycle dynamics. And so here we had access to sort of this Fucci reporter that kind of broke cells down into two distinct bits of the cell cycle, and that allowed us to show that there was at least 2 checkpoints that are mechanically regulated. And along the way, and I can talk more about this more too, I tried to highlight that experimental design really matters, and I'm going to come back to both these points in a minute. And so it's important we think about the limitations of data, not only when we build the model, but also in the inferences we draw from it. Okay, so why do I say that? So we parameterized our model using these kinds of data, and they're actually data from a cell line known as an MDCK cell line. It doesn't particularly matter what it is. But I want to just highlight that this cell line is precisely the same cell line as was used in that Stryken paper that found one of these cell cycle checkpoints. And one of the nice things you can do is then take this model and take the parameters from this model and use it to kind of simulate or predict the dynamics of those experiments by Stryken et al. Now, these are actually, and it doesn't look like it at all, these are exactly the identical experiments. They were barrier assays. Cells were grown inside this cookie cutter. But what Stryken et al. Did was they grew them like to a really, really high density before they took the barrier away. And so all of the cells you can see here really are essentially stuck in G1 or G0. They're arrested probably, right? And so the impact of that is that when you remove the barrier at time 0, you've pretty much only got, like I said, cells that are in G1 that are red. And even after 15 hours, cells have begun to move and they've begun to fill the available space, but we really haven't seen cells kind of exit sort of G1 and move into SG2M. And it's only by about 30 hours, well, maybe a bit before, that you can see that. But with the right initial conditions, our model is picking that up really nicely, and it's a really nice prediction of what happens, which is great. So you know, we can transfer our model to different scenarios. And we could also transfer our model to the scratch assay data that I showed you right at the start. And in fact, if you do that, you get a very good prediction of how the space is filled by these invading cells. But the other thing is that you can get just as good a fit by considering this model that doesn't have any of that cell cycle regulation in it. And really the reason for that actually is that the cell density is so low, right? I didn't really refer to this the first time around, but you know, we've only really working in a context where about 15% of the available space is filled. And so those feedback terms that are higher order are just not important in capturing the dynamics. And in fact, everything that I've talked to you about today, right, came from the fact that the first time we worked with this data set, we non-dimensionalized everything with the confluent cell density, which removes those capital K1 and capital K2 parameters from the model. And so we never tried to estimate them. And if we had, what we would have found is that they were completely unidentifiable from the model, and that is the model that we should have been working with. So hindsight, I guess, is always 20/20. But it was certainly something that was useful to think about sort of after we developed our model. And then just briefly looking forward, Martin Glerich's group, who sort of provided evidence for that other checkpoint, have done a range of experiments with now what we call four-color Fucci. So we were talking about this at lunch, about the fact that kind of where things go in biology is always driven really by the technology that's available. Well, the new technology now is four-color, and I think even more colors that represent or break down the cell cycle. So one of the things I'm quite interested in doing is thinking about whether our approaches carry over. I mean, in the obvious sense, they do, right? We can just roll this out and apply it to data from a four-color Fucci where we can see all four phases of the cell cycle. But I'm reasonably confident that things aren't going to be as easy because we had this factor of 2 in one of our equations before, which came from cells dividing. And when we write down a four-color model, we're going to have 4 equations, and the factor of 2 is only going to be in one of them, and I think that's going to absolutely cause us more pain than we might hope. So anyway, maybe at some point I'll be able to tell you what happens with that. So I'm just going to conclude now by acknowledging people who did a lot of this work. So Carlos Falco is an incredibly talented PhD student who's just graduated from my group, and he did lots of the applied work. Alex Browning was a key collaborator on the structural identifiability work, and then Kevin and Daniel generated lots of the data. And if you're interested in more about the ideas of identifiability, there's a preprint there. So with that, I'm going to say thanks a lot for your attention, and I'm happy to take any questions.